Welcome to the second part of this Chapter 9 lecture. So before we can really get into the breeding aspect of crop plants, we have to look at a couple different kind of definitions and how we utilize all of them in terms of plant breeding. So first thing that we're going to look at is a genotype versus a phenotype. And for some of you, this might be a little bit of a review, but that's okay. So the genotype, when you think of think of the all of the genetics. So total genetic makeup. So it has to do with everything genetic about that plant. The phenotype, though, is due to the genotype. So it then takes the environment and their reaction to the environment and two different things having to do with that, the growth of that plant, the growth and the development of that plant. So genotype, very broad, okay? Think the genetic makeup. Phenotype has to do with the genotype. So G is before P. So G equals genetic makeup. Phenotype is also because of the genetic makeup, has to have that phenotype, and then has to work within the environment as well. The heritability, the qualitative traits and the quantitative traits then come after the genotype and the phenotype. So the heritability, the proportion of that phenotype, that's controlled by genetic factors. Again, going back up to that genotype. Quality traits, we have to look at one or a few of those genes. They're very discrete, and when you're thinking quality, you're thinking just one or a couple. You're not thinking a lot. The quantitative traits, so we're looking at a lot more different traits, and they're more complex, and we breed them on a larger scale. So qualitative, quantitative, and then heritability then have to do with the genotype and the phenotype once they are then formed. So Mendelian genetics. So I'm sure you all have heard of Mendelian and the pea pod plants and how they're really important to crop science and livestock, to be honest with you, and how we breed both plants and livestock. But you know, it's pretty interesting. I always, when I learned about him in my school years, he was an Australian monk, and that was really cool to me growing up. He was, you know, he wasn't just a scientist. He had other aspirations, and he did other things with his life. And so he came up with this pea pod experiment, and it kind of skyrocketed a couple hundred years after that. Not really when he discovered it, but I think it was 1865, reported that there was he predicted a pretty intense visual comparison or appearance of the pea plants because of the way he had bred them. So we really look to him as kind of the grandfather of how we breed plants and livestock because of what we call the laws of inheritance. And so how the predictability of that behavior of those chromosomes then transforms those genes and those parents to an offspring. And so really important in terms of how we breed plants today. So we look at three really intact factors of Mendelian genetics. We have to look at the characters that are controlled by the two allies. We look at one ally and how it's determined it determines the phenotype observed for that trait. Now remember, phenotype is due to that genotype. So we have to have that genotype first, which is very important with that first factor. Okay, so then the third factor, one of those chromosomes from that parent will then be passed on at random to each of those gametes. And we'll talk about in class this week how important it is to kind of know a pretty basic form of statistics. So in plant breeding, how you are statistically analyzing each of these inheritance and chromosomes and how they will then be pushed off into a plant breeding aspect. So I'm sure you've seen this. This is that cross of those pea plants. So we have the homozygous versus the heterozygous, and you can see here the homozygous dominant and then crossed with the homozygous recessive gene. And when we do that, what are we going to get? Okay, we're going to get that. We're going to get that F1 gene. We're going to get that that green pod. But when we cross the heterozygous dominant by the homozygous recessive, we're going to get 2 and 2. So it's kind of at random. And I want you to make sure that you have read over this chapter, because we're going to do a lot of these Punnett squares in class this week, so that you can really understand and know how to do a Punnett square, because it's really important, not just in plant genetics, but also in livestock genetics as well. And we've used this, pretty old way of doing inheritance for a long time. So it's obviously worked, and that's kind of how we do it today. So make sure that you kind of understand or have at least read it, so that we can do the fun activity that we have planned with real pea plants, snap peas, this week. So a couple more goals, or a couple more definitions that I want you to know. A lot of people think that yield, when we talk about yield, it's the main goal of how we look at plants. But in some cases it is. It's that quantitative trait. So we want the biggest yield. It would be incredible to grow 200 bushel corn in Oklahoma. I mean, that would be awesome. But that's yield, right? But is that also then going to compare to the disease, the insect resistance, the drought tolerance, how competitive it is with not only other plants around them, but also the weeds as well? Okay, so yield, very complex, quantitative, so we're counting the numbers trait. Quality trait, so we're looking at this has a lot to do with like forages, so the digestibility of those, the quality traits, how it's fermented, is it quality for that animal? Okay, is that hay quality for that animal? Harvestability, we're looking at specific crop loss in terms of how we grow plants. Persistence, we're looking at how that survivability will happen long term. So is it going to be able to survive a drought? Is it going to be able to survive 8 inches of rain in 3 days? Is it especially after you plant it? So what is that persistence level at, and how can I make it better in terms of plant breeding? So let's look at the difference between qualitative and quantitative traits. Qualitative, again, I'm going to separate it into distinct classes, and it's only going to be looked at by one or two, maybe 3 genes. So a good example of that would be those green or yellow pea pods that we've talked about, and we'll look a little bit more into this week in class. The quantitative, it's really hard to separate them into different classes because of how many genes it takes to do that. And then a good example of that is the height and the rate of that plant. Okay, so you can go out when we're looking at the bean trials this fall. You'll be able to go and see the different height, the growth, how the yield will be a big one in that quantitative trait. But the quality traits, you're going to be able to see differences in color. You'll be able to see differences within yield too, but how that growth and development is then taking place within that bean plant. So again, we've talked about how yield is important for quantitative traits, and how that quality trait will then be important within digestibility within that plant, and how all of that is very important. Harvestability, we've looked at persistence, especially in perennial crops. Those are all very, very important. Hint, hint, you're going to need to know these. That's why I've said them twice. So what are some strategies within plant breeding? Okay, the first one we have to look at is collecting all of those genetic variations. So I'm going to be able to show you, we're going to be able to look at this when we look at the soybean plants, or the soybean trials that we have at the GRDA research farm, and we're going to be able to see the genetic variations from many different varieties, because we've determined different parent materials. We've looked at different genetic or new genetic combinations. We've looked at the selection of different combinations of traits. Then we've made that evaluation trial that seed has made it to that new variety that we have named, and that's what you're going to be seeing in the field this fall. And then after we have discovered, okay, that new variety, this variety it's grown, we can make a yield out of it. It's in some cases it's drought resistant. It's resistant to some pesticide or to some insects. It's good for farmers to buy, and they're actually going to make a little bit of a profit or break even. Then we will distribute it to the growers. So that's the big part of why we utilize these evaluation trials, and then look at the varieties so that farmers and ranchers can come and look at them to see what they would want to grow. Those different scientists at different universities or with different seed consultants can say, okay, this different variety grows really well in clay soil. This variety grows well in silt loam soil. So you can kind of mix and match what you would rather have in your field, which is awesome because you're the person that's making those decisions. So you want to make sure it's the best decision for not only the climate, the weather, the soil, and all of those in your pocketbook, how much it's going to cost. Those are very important to how we look at strategies of plant breeding. So let's go back to self-pollinated versus cross-pollinated. So we've talked about kind of the differences of both and why we utilize both of them, but I want to kind of break down why these are very important. So you look at self-pollinated, they're usually, almost always homozygous, and they're a pretty common method of breeding that will then give you a pedigree selection or a mass selection that you can look at and you put pencil to paper and kind of make your selection of that. Cross-pollinated is heterozygous always. It can have a mass selection, although sometimes it's not very known. And the hybrid is most commonly used. So the hybrid that we then have bred is most commonly used. So if you've read, you know that the F1 generation is important within the pedigree selection. So we have to look at the F1 generation that is planted and how its superior plants are then selected. So we're taking out the F1 plants that are actually doing well. They are doing well within the climate and the weather, whether it's a drought or there's too much water, whatever that might be, it's doing well within that climate. Then we're going to take those plants and we're going to collect those the seeds from those plants, and then the undesirable plants or the plants that might not come up to par will then be thrown away and we won't use those. Then we will select different rows within those different varieties and select those additional plants for breeding, and we can do that up to 6 times. So F1 is that first generation, and then F6 is that 6th generation, and then at that F6 generation, those plant rows will then become very uniform. They will look almost exactly alike. Those seeds will then be selected within rows and then bulked, and then we'll look at the yield trials within them, and those are done all throughout the world, and it's really neat on a producer basis to go see them in different parts of even the 4-state area or even Oklahoma, because people can grow soybeans way differently in southwestern Oklahoma than we grow it here, just because of the climate difference that we have 5 hours southwest of us. So that, making sure that you're doing your homework on those yield trials and evaluating those that are close to you is really important. So mass selection, what is it? Why do we use it? Mass selection is important because we look at the desired characteristics and that are selected. And so those plants are then selected in bulk and used to grow the next generation. So again, you might be getting a lot of different genes, but you're only specifically picking for one gene. So it's on a mass portion. Then it's repeated for a lot more generations, about 3 or 4, maybe 5 generations. So the protein content could be a really good example of that. We're increasing the protein content, so we're going to select about 5% of that selection intensity of that mass selection, and then putting it into the plant and to breed it so that we can have a bigger protein content. This is really common in soybeans.